Hello everyone, I'm your host Lokendra Kumar, and today what we will be discussing is the topic named Microbial-Associated Molecular Patterns, or in short it's also known as MAMPs. Before I start this video, I have a quick request to make. If you are new to the channel, then please support us, subscribe to the channel, and if you want to see similar kind of videos, you can also type in the comment section and let me know if there is a specific topic that you want to understand. I can create videos on those specific topics. And for today's video, I have MAMPs. I've created one video, doodly video on this. Probably you have seen that video. That video is a summary of what we are going to discuss today. So we'll discuss all those points in detail. This is more like the lecture-based video, and that is more very quick snapshot of the MAMPs in general. So you can see overall everything is being laid out over here. We will be understanding what are MAMPs, what are the different types of MAMPs, what are their roles, and also what is the significance or the importance of MAMPs in general when it comes to gut health, when it comes to human health. So you can see here keywords are also written. But let's start the video. And the first point that I would like to make is what are microbial associated molecular patterns. That is a very, very important point. So what are the molecules that are known to be MAMPs when it comes to microbes? And let me give you a little bit background on this one. So as you can see over here, what we're discussing is one of the topic in microbiology, and we have a very close association with microbes. We live very closely with microbes. We live in association with microbes. Microbes are important for our health. Microbes are beneficial. They are also harmful. And harmful microbes, they're going to cause disease in our body, and then beneficial microbes are going to give benefit to the body. So it's a kind of very, very balanced relationship when it comes to the host microbe interaction. So in that case, you will have beneficial microbes, you will have harmful microbes, and then both of them will have molecules that will interact with the host. Those molecules that interacts with the host are the microbial-associated molecular patterns. Now you can see the word microbial-associated molecular patterns. It is again self-explanatory. It can explain itself, as you can see this entire title. So all the patterns, and patterns we are referring over here are the molecules. So let me write it down over here. So molecules basically. Molecules, right? I need to practice more on this thing. Molecules, and those molecules that are associated with microbes, right? That is why we call microbial associated molecular patterns. They are present in large number. They form a specific pattern on the surface or inside the microbe, and then they are associated with the microbes. That is why we call them microbial associated molecular patterns, or MAMPs. MAMPs, right? MAMPs. Another word is also being used, pathogen-associated molecular patterns. So those are specific to the pathogens, and there is a kind of overlap between these two because they are present in both of the organisms, and it's kind of very interesting classification, right? When it comes to pathogen-associated molecular patterns, we call PAMPs, and then you have receptors to detect those PAMPs, right? And in case of in general, if you want to call them. Both in case of pathogenic as well as in non-pathogenic or beneficial microbes, you have the term with microbial-associated molecular patterns that become more general. Correct? Okay, so this is one point, right? Let's move on to the next one. So what are those molecules, and how we define these microbial-associated molecular patterns? Let's try to understand that. So I have this first slide where what I'm explaining is these are molecules. Now when it comes to the word molecule, that means they are not live organisms or organelles. They are basically the parts of the microbes, and they are small molecules. So MAMPs or PAMPs, they are these small molecules. They can be larger molecules, so they are not just small molecules because the classification is huge. So you have the molecules that you characterize as MAMPs. They can be protein molecules, they can be carbohydrate molecules, they can be lipid molecules, right? So they are all molecules associated with the microbes. Next is they are found on the surface. Majority of the molecules, they are present on the surface of the microorganisms, and that is how they interact with the immune system, the components of the immune system. And I have this illustration. I hope I should be able to explain few points over here. Right, it's very difficult. I'm trying to write things. Okay, maybe slowly I'll get used to it, right? Okay, I have this illustration that I've designed, and I have some round structures and also one hair-like structure over here. What is this? This is a bacterial cell that I've created over here, and you can see this is the surface, and on the surface you can see the molecules, and we can say these are MAMPs. I'm just writing over here. MAMPs, right? It can also be another. This is MAMP1. This is again another MAMP, which we will understand. This is flagellum, and the protein present inside the flagellum is flagellin. That is another molecule that is characterized as MAMPs. And then you have LPS. So I'll just write flagellum. Why don't I write flagellum over here? Right. So I have a kind of running handwriting. I hope you are able to understand what I'm writing. So LPS, lipopolysaccharide. So it can have lipopolysaccharide. It can have peptidoglycans. So these are the major MAMPs that we are going to understand in this one. There are other molecules associated with MAMPs. This research is also going on to understand what are the additional MAMPs that are present. So two important points over here. One, they are molecules. They can be small molecule, they can be larger molecule. Most of them, they are larger biomolecules. They are present on the surface of the microorganism. Three important MAMPs that are well characterized: peptidoglycan, you have LPS, and flagellum. They are present on the surface of the microorganisms, and they are present in beneficial as well as in the harmful. So when it comes to, this is also a very important point, and I think about these things a lot. What is the difference between the pathogenic peptidoglycan and the sorry, the harmful microbe peptidoglycan? There should be subtle difference because they are going to play a significant role in their interaction with the host. And then when it comes to LPS, interestingly, research have shown that there is research, various research studies, they have shown that there is a difference between the LPS of pathogenic microorganism as compared to the beneficial microorganism. But when it comes to pure LPS, and if you extract it from beneficial even, like non-harmful E. coli, it can induce a potent inflammatory response. So I think it will also depend upon the amount and type of these molecules that are present in the beneficial versus the harmful organism. So it's a very important topic to study or do research more and understand how they interact with the host. And a lot of studies, a lot of people are working in this area. I'm also working in this area. That I'm trying to understand how these molecules, and not only these molecules, there are other molecules, how they interact with the host, and how they help microbes in providing benefits as well as in providing the toxic effects to the host, right? So two important points. We covered molecules. They are molecules, biomolecules. They are present on the surface. Let's move on to the second one. So right now what we are doing is we are trying to understand the basics of MAMPs. Second is they are recognized by the host immune system. That is very important point. And I hope everyone understands. Those who are watching my videos, I've made multiple videos on immune system. We have two types of immune system. Let me write it over here. One is, I don't know why I'm writing adaptive. Let's write adaptive first. And the second one is innate, right? So what is the basic difference between these two? One is very non-specific, innate, right? It's non-specific. Anything that is harmful, it's try to block it without understanding what that agent is. But adaptive is it generates when that particular specific organism enters in the body and interacts with the host cell. So you can see over here what I've designed as some of the components of our immune system, the immune cells, right? So just imagine immune cells, and obviously they are not like this, they are just illustrations. So you have immune cell 1 and immune cell, second immune cell over here. So what they are trying to do is when there is entry of these pathogenic organisms, let's see, the pathogenic organism is able to enter inside the host. It is able to breach the innate barrier, and then what will happen? It can first interact with the cells of innate immunity, which are non-specific, which are going to provide non-specific immunity. So we have macrophages. You have other polymorphonuclear cells. They will interact with the organism, and they are trying to understand the molecular patterns that are present. And because of these molecular patterns, there will be interaction with the host receptor, and based on that, the immune response will be generated. So they are basically recognized by the immune system. That is very important point. And second one is they're able to stimulate the immune system. Now, when it comes to stimulation, there should be the interaction between the microbes and the immune cells. And there are molecules that are going to induce that interaction. In microbes, you have MAMPs, and in case of the host, you have receptors, protein receptors. So those receptors should be able to interact with the MAMPs molecules. So one of the important family of those kind of receptors are toll-like receptors. I have made multiple videos also on toll-like receptors. That is also one of my research area. What are toll-like receptor and why they are called toll-like receptor is very important, because they are the cross-checker, right? The word toll come from whenever you have a checking system. So they check if there is anything that is not from the host body. Their role is to recognize that and response against that. So it stimulates the immune system, and all these molecules, they are able to stimulate the immune system, right? So that is another point. So first point is they are present on the surface, majority of them. A lot of research is going on, and second one is they are able to stimulate the immune system. Let's move on to the next one is essential for maintaining gut microbiome. Yeah, this is something people are doing a lot of research on, gut microbiome. The microbiome present in our gut, although microbiome is also present in lung, microbiome is also present in oral cavity, on skin, it's present on the gut. And inside the gut, you have huge amount of microbes. So this is just imagine this, because these are all illustrations, right? This is the gut. And inside the gut, you see these microbes. And here, why the microbes are present here? Because high nutrient conditions are there, and it's also in direct contact with the environment. This is the alimentary canal. You eat the food, it will come with the microbes, and then it's not going to be 100% sterile, right? So it will come with the microbes, although stomach is going to take care of many of the microbes because of the acidic pH, but still some organisms, they will go into the system of the basically intestinal environment, and their chances are, if they are beneficial, then it's fine. If they are harmful, then it's going to cause the disease. Beneficial means you take probiotics, right? And then they establish over there and then provide you the benefit effects. So in case of beneficial microbes, so that is why we are not using the word pathogenic associated molecular patterns, because they are going to stimulate the immune system. They are going to stimulate in a way, they are going to establish themselves. Here, they are going to help the gut microbiome, and the beneficial microbiome are going to be in high amount, so that the host gets the benefit. They prevent the growth of pathogenic bacteria. So yeah, this is something, it's very interesting, because LPS is also present in pathogenic bacteria. LPS is present in beneficial bacteria, but in case of peptidoglycan also. But when it comes to the slow stimulation of the immune system by the beneficial microbes, what it does is basically it helps the gut microbiome to get rid of the pathogenic bacteria. So that is why second point is it prevents the growth of the pathogenic bacteria. It's a very important point, right? So these are some of the key points regarding what are MAMPs. Now moving on to the examples of MAMPs, you have three important molecules. First one is LPS, lipopolysaccharide. It's present in gram-negative bacteria. Second one is peptidoglycan. Third one is flagellin. These are the schematic I've designed. This is basically the schematic structure of endotoxin. LPS is also known as endotoxin, and lipid A component of endotoxin. I'll explain it later on. This is peptidoglycan. It's made up of sugars, and then you have flagella. This structure, not this one. This is the flagella, and the flagellin is the protein present inside the flagella. Okay, let's understand all these MAMPs one by one. LPS, it's also known as endotoxin. I already told you. It's present on the outer membrane of gram-negative bacteria only. Remember that. It's not present in gram-positive bacteria. It's present in gram-negative bacteria. And it's highly potent stimulator of the immune system. So that is the second point. It stimulates the immune system very, very quickly in very, very small concentration. It causes inflammation. So if you have LPS from gram-negative bacteria in high amount, it enters into the bloodstream. It will immediately cause the inflammatory reaction, release of inflammatory mediators, and leading to the potent inflammation. It's also known as I already told you, endotoxin, right? So, when it comes to endotoxin, when it comes to exotoxin, there are a few points that you need to understand. Why don't we call it as an exotoxin? Why other molecules are known as exotoxins? Exotoxin is something that is secreted by the bacteria out. Endotoxin is something that is present in the bacteria, right? So endotoxin is present in the bacteria. It will released out when there is a damage to the bacterial cell membrane. So it's an integral part of the bacteria. Okay, exotoxin will be something that will be secreted out. Anyways, I hope now the point is clear, right? Next point regarding the LPS is composed of two important parts. One is lipid A moiety, and second one is core oligosaccharide. Sugars are present. Now, it's interestingly, it's made up of, suppose this is your lipid A, and then you have sugar molecules like attached to this, and the size of these sugar molecules will vary. And it's also going to change depending upon the strains. It's also going to change depending upon the bacteria. And serotyping is also being done because of this. It's also known as O antigen, right? So you have two important points, that is parts. One is lipid A and then the sugar moiety. That sugar moiety is non-toxic. Understand that. You have this lipid A moiety, and this is the sugar moiety. Sugar one is the non-toxic part, and it is also, as I told you, immunogenic. So if you have antibodies against this, it will interact and specifically bind to those molecules. Lipid A is very, very toxic in nature. It is going to cause potent damage into the host. So all the toxic effects of the LPS is because of the lipid A, and the immunogenic effects are because of your sugar moiety, right? So this is the general structure of LPS. Next is peptidoglycan. So I'm just creating various sugars, and peptidoglycan is made up of different types of sugars. Now you have the molecule that is present on the cell wall. It is present on the cell wall of the gram-positive and gram-negative bacteria. In gram-positive, you have high amount of peptidoglycan. So that is why the cell wall of positive bacteria is really thick. It's very difficult to break down the cell wall of gram-positive bacteria as compared to the gram-negative bacteria. Next is, it is less potent stimulator of the immune system. That you have to understand. So because of this one, I think it can be a very important candidate also for the stimulation of the gut in a healthy way. So if you have the peptidoglycan of the beneficial bacteria, that can be used as a stimulant of the gut in a moderate way. Role in maintaining gut health. So people have also done a lot of research on the role of peptidoglycan in gut health. It also maintains the structural integrity of the bacterial cell and also the shape. As I already told you, that lipid layer is not going to help in the structural integrity. So if you remember, we have two types of cells: gram-positive, gram-negative. In gram-negative, you have three layers. The central layer, this one is the peptidoglycan, and it's thin. But in case of positive, you have a very thick peptidoglycan layer, and then you have lipid layer. Lipid layer is also present in case of gram-positive. So this is positive, this is negative. Right here, you have very, very thick, and then here this is relatively thin in case of gram-negative bacteria, right? And because of that, in both cases, the structural integrity, because this is a tough molecule, this is a tough biopolymer, so it's going to provide the structural integrity. It's going to help in the shape of the bacteria. It also protects the bacteria from osmotic stress. Yes, obviously, because this is a kind of wall, outer wall of the bacteria. So if there is any osmotic change or any other environmental change, that this molecule is going to help the bacteria to take care of that change, right? Okay, next. Few other points. It helps in cell division. So a lot of research is going into this one also, and there are so many molecules are being developed that can target the peptidoglycan layer, so that we can kill pathogenic organisms like Staphylococcus aureus. It also determines the shape of the bacteria. Again, another point is antimicrobial peptides. AMPs. I made one video on AMPs. So AMPs, if you want to see that video, just type antimicrobial peptide basic science series. You will understand what are AMPs and what are their roles. Peptidoglycan helps in the formation of or the production of these AMPs in the gut. So it helps in the defense against the pathogenic bacteria, because these AMPs they have the potent activity against pathogenic organisms, and they can also modulate the gut microbes. This is how they're going to modulate it, because they can reduce the population of pathogenic organisms, and then they are not going to harm the beneficial microbes, right? These antimicrobial peptides. They are very specific, some of them. And a lot of research is going into this area to develop new antimicrobial peptides. Next, one of the important molecule is flagellin, flagella. The molecule that is used for bacterial motility, movement in liquid environment. So bacteria can basically swim through liquid environment. So I hope I'm creating illustration that you can imagine the bacteria is swimming in the liquid environment, right? So that will help you to understand that how it is basically helps the bacteria survive in the liquid environment. So it can move through the liquid environment. And what are the role of flagellin? One point is it's a protein molecule, right? We talked about other molecule, lipid molecule, carbohydrate that are present in the lipopolysaccharide, and then you have protein molecule. This is a protein molecule, and it's a potent stimulator. So among these three, two are potent stimulators. They are really potent stimulators. One LPS, and second one is flagellin. And less potent stimulator, second one was the peptidoglycan. It helps in bacterial motility. I think that I've explained with this illustration. And then bacteria can swim through the liquid environments. So bacteria can swim if the environment is liquid. And this protein is not present in all the bacteria. Only motile bacteria will have that, pathogenic as well as non-pathogenic. And comparative analysis will be very, very interesting of the flagellin protein of the pathogenic as well as the beneficial bacteria. Host defense mechanisms, because of its ability to induce the immune response, it can help in the defense mechanisms, right? Okay, what are the other points regarding the flagellin? Flagellin interacts with the toll-like receptor 5. I don't know if I remember I discussed the toll-like receptor 4. For LPS, you have toll-like receptor 4. For flagellin, you have toll-like receptor 5. And there are other toll-like receptors like TLR2 is there, TLR7 is there. I think 7.1 is for the genetic material. Anyway, so other like toll-like receptor 2 is there that will interact with the lipoproteins. You have toll-like receptor 5, which will interact with flagellin. So that is known. Our body will have toll-like receptor 5. So basically, flagellin will interact with TLR5. Okay. Because of this interaction, it will induce the series of signaling events that will cause the release of inflammatory mediators and also activates the immune cells. And a lot of research is going on further to uncover if there are any other microbial-associated molecular patterns that are specific to pathogens, that are specific to beneficial microbes. What are their roles? What are their roles, especially in human health? What are their roles, especially in other diseases, other metabolic diseases, right? It's very interesting study. The topic is very, very interesting to understand how the microbes, they interact with the host and how the interaction will lead to the understanding of the molecular mechanisms. Okay, why they are important, right? What is the significance? Significance. Probably, I think I'm getting better in writing, right? So I think this is significance. So yeah, spelling is correct. So what is the significance of these MAMPs? I have written all the points that we have already discussed. So let's summarize these. One is the stimulation of the immune system, right? So if I again show you with the illustration, suppose this is the immune cell, correct? So this is the immune cell. And the receptors, they are present on the surface of these cells. Now microbes will be there and they will interact, and then because of that, they will cause the release of inflammatory mediators, right? And then that will cause the stimulation of the immune system. Next is prevent the growth of pathogenic bacteria. The pathogenic bacteria that can cause disease, they can prevent the growth of those. Antimicrobial substances, we have already talked about antimicrobial peptides. These can directly kill the pathogenic organisms. They can help in maintaining the structural integrity of the intestinal barrier. So they can also help in maintaining the structural integrity of our intestine, and how the research have shown that the production of tight junction proteins. These are the proteins that help in the maintaining the integrity of the intestinal barrier. So this is how they can basically help in maintaining. For example, just see you have this one cell in the intestine, and then you have the second cell, and then these cells they are they have these tight junction proteins. And these proteins are important because if they are present and they are integrated, what will happen? The cells, they are going to be tightly, bound together. So that will help in overall maintaining the structural integrity of the intestine, right? So I think that is all. We have covered so many things. These are some of the references that I recommend, and I have also used these references to create this this detailed video. The first one you can see is the gut microbe and the liver cancer. So this paper basically will help you to understand what is the role of gut microbe and the liver cancer, and it will also tell you a lot of mechanism associated with the gut microbes and what are the molecules that are associated with the interaction of the host and leading to the cancer, right? Next is new piece in understanding hepatocarcinogenesis. It's also especially in the context of gut microbiota. So focusing on gut microbiota, how it can induce the cancer, because most of these articles will be related to the signaling pathways, especially in cancer. Another one is, you can see over here, in associated with plant immunity. I think I wanted to also put another article which is not just with the human associated with human immunity, but it can also modulate plant immunity. Very interesting article, right? Next, you have Associated Molecular Patterns in innate immunity. So I think this one is also very, very interesting. There are so many articles. These are few of them, right? I'm not promoting anything, but when it comes to research, everything is basically for public information. So you can read other articles also. When you open these articles out, you will see a lot of references in these articles, and you can also find out the suitable one for your research, right? So you can read them and understand molecular patterns in more detail. Correct? Those who are researchers, they understand what I'm saying. References are very, very important. References are critical, especially when it comes to the research as well as teaching, right? Okay, so I think that is all. I have, I think, summarized the MAMPs in detail. Those who are, more interested in this topic, I'll be creating more videos on these specific molecules, very, very detailed videos. We'll go into molecular details and the mechanism of these molecules, and we can also see what's the latest research saying about these molecules. It will be very, very interesting. And second, if you like the content that I create, then please do support the channel. And how you can support the channel is, if you have watched the video and if you like the video, then yes, hit the like button and do watch the videos, the full videos. Yes, obviously they are teaching based videos. They are not very, very entertaining kind of videos. They are informative videos. So you have to spare some time. You have to make up your mind. You can't just be doing social media work and then you're also watching the videos. You have to think in your head that, okay, I'm going to watch one basic science series videos. Then I have to spend significant amount of time, and I have to make sure that, okay, I'm watching something which is very, very more information kind of packed videos, right? So watch the entire video, and if you like the content, then now please hit the like button and subscribe to the channel. Share the content with your friends, with your in your social media platforms. So it also helps the content creator to reach out more people, and then also benefit all those people if they are interested in these topics, right? So I think that is all for today. I hope you were able to understand MAMPs. What we have covered is what are MAMPs, different examples, and also how these MAMPs, they interact with the host, and finally, what are the key benefits. I'll be bringing more videos. I'll be creating short, concise videos. I'll be creating more lecture-based video. I'll be creating more illustration-based videos. I'll be creating more tutorial-based videos. So please stay tuned, and we will be doing a lot of interesting interactions on all those topics. If you have doubts, if you have any questions, you can post those questions in the comment section. I'll make separate videos on the comments that you guys post, and I hope in future also I'll be creating more videos that are going to help you in your studies and in your exams. All right, till then, take care and study well.